The overall goal of this procedure is to efficiently isolate protein complexes from mammalian cells. This is accomplished by first transfecting the cells with a HaloTag fusion construct to express the protein of interest. The second step is to lyse the cells and covalently capture the protein complexes on resin via HaloTag. Next, the protein complexes on resin are gently washed to remove any nonspecific interactions. The final step is to elute the protein complexes from the resin. Ultimately, HaloTag pull-downs are used to isolate and discover novel protein interactions from mammalian systems.
The methods we will demonstrate here to you today can enable key discoveries in the area of functional proteomics, including identification of novel protein interactions and determination of protein localization inside cells. Performing these procedures today are two of our senior scientists, Jacqui Méndez and Hélène Benink.
First, for each fusion or control, prepare one 15-centimeter dish with 30 milliliters of cells at 3 to 4 times 10 to the 5th cells per milliliter, or 1 to 1.2 times 10 to the 7th cells total per construct. Incubate the cells for 18 to 24 hours at 37 degrees Celsius and 5% CO2. Following incubation, transfect the construct with the desired transfection reagent.